Welcome back, class. Let's take a closer look at the nephron of the kidney. And each kidney's got millions of nephrons, the functional unit of the kidney. So if we look at the lower left, we know that the outer part of the kidney is called the cortex. And we know that the inner part is called the medulla. So we take this section here, we're going to blow it up. This part here is all the renal cortex, and the dark stuff here is the renal medulla. And everything that we're seeing in yellow here, this is all the different parts of the nephron. They call them the tubules. And let's review some of the main parts of the tubules. So right here, let's say this one will put AA for afferent arteriole, and then we'll put efferent arteriole here. Afferent arteriole brings the blood into the glomerulus. The glomerulus is stored in Bowman's capsule. You're going to get debris and things that your body may want to excrete from the body. Whatever it pulls into the first part of the tubule is called filtrate. So the filtrate is pulled in to the proximal convoluted tubule. And it's pretty convoluted. Now it's going to drop through the cortex into the medulla. That's the loop of Henle. The loop has a descending limb and it has an ascending limb. This is the loop. The descending limb is bringing it down. The ascending limb is bringing the filtrate up. And now we're back in the cortex again for the distal convoluted tubule. Now, I want to point something out to you. Notice that this part of the distal convoluted tubule is very, very close to the afferent and efferent arterioles. So there's lots of cells within this area here that's responsible for detecting what the blood pressure is like and what the oxygenation is like within the red blood cells. And this region right here, they call it the juxtaglomerular complex. Juxtaglomerular means just next to the glomerulus. Juxtaglomerular complex. And there are cells here that are responsible for releasing renin and erythropoietin. That's the region right here that does that. And it's part of that distal convoluted tubule section that's very, very close to the glomerulus again, afferent and efferent arterioles. Okay, after the DCT, the distal convoluted tubule, we're now into the collecting tubule. These are collecting ducts. These are collecting ducts. All the collecting ducts lead into the collecting tubule, which leads into the ureter. Okay, now here are the basic processes that take place in the nephron. So the kidney carries out three basic processes, and the sum of these processes are going to determine the rate of excretion. So with the exception of proteins, most substances in the plasma are filtered and entered the Bowman capsule. So here's your afferent arteriole, here's your glomerulus, and here's your Bowman's capsule. The first part we know is going to be the proximal convoluted tubule. So number one is filtering that can take place. So we got some filtering. Now maybe there's some water, maybe there's salt, maybe there's glucose, maybe there's amino acids, maybe there's stuff that's being purified and cleaned out that the body's like, hey, I don't want to get rid of this. we need to get that back into circulation. So number two, it's going to get reabsorbed. And whatever the body doesn't want to reabsorb, it'll push back out. That's when it secretes. And after it's secreted, it can be excreted. Okay? So you have filtration, reabsorption, and secretion. And after it's secreted, your body will urinate it out, which is part of excretion. And again, this picture just depicts everything from that arcuate artery to the cortical radiate artery to the afferent arteriole to the glomerulus. And it's not the best to really show proximal convoluted tube. It's so convoluted and obscured, so it's tough to differentiate. I mean, they do label it, but you really have to follow it really, really close. Okay. So the glomerulus, that's a capillary bed. It's attached to arterioles on both sides to maintain high pressure. The large one is the afferent arteriole, and the narrow one is the efferent. And we said that blood comes in this way at a faster rate than it can exit, so it stagnates here for a little bit, and it allows for the pulling out of all the debris, right? The filtration. It can filter and pull it out into the first part of the proximal convoluted tubule. And this part has simple cuboidal epithelium. The proximal convoluted tubules and the distal convoluted tubules both have simple cuboidal epithelium. You can see these are cube shaped. Simple cuboidal, one cell layer thick. Notice what surrounds the glomerulus are cells called podocytes. And these podocytes create little filtration slits. And these filtration slits only allow certain size particles to be filtered out, right? It doesn't want to allow proteins out of here. It doesn't want to allow full white blood cells out through here, right? If you're seeing white blood cells in urine, there's a problem. If you're seeing protein in urine, there's a problem. If you're seeing sugar in urine, there's a problem. That's why we do these urine analysis tests to pick up these things. If we see red blood cells or white blood cells, there's a problem. We don't want to see those there. So the glomerulus sits within the glomerular capsule, and the glomerular capsule, that's called the Bowman's capsule, same thing. So you've got your Bowman's capsule, which is this. You got your glomerulus. One of these will be the afferent arteriole and one will be the efferent. Then you have your proximal convoluted tubule. Then you have your descending limb and your ascending limb of the loop of Henle. So that is the loop of Henle. As it goes down, that's the descending limb, and that's the ascending limb. Notice this is thinner and this is thicker. And then you're at the distal convoluted tubule, all parts of the nephron. Okay, another good picture, very clear to show the arcuate artery that goes along the base of the pyramid. Off the arcuate is the interlobular. Remember, the other name for that is the cortical radiate artery. Off the cortical radiate, that's your afferent arteriole, large diameter blood vessel. Efferent is coming out. And you can see, look at all the blood vessels surrounding the tubes. And that's why you can have filtration, reabsorption, and secretion, which eventually leads to secretion. Remember, this is a two-way street. Things can go from tubules to blood or blood to tubules. All right, so here's the glomerulus. There's your Bowman's capsule. We follow this. This is the first part. This is your proximal convoluted tubule. And we follow it down here. It's coming down, that's your descending limb, loop of Henle, ascending limb, distal convoluted tubule. Now look, the distal convoluted tubule gets very, very close to the afferent arteriole. These are the cells of the juxtaglomerular complex, right there. It can tell what's going on with the oxygenation, the blood pressure, and everything as it's coming in. So these JG cells are really important for renin production and erythropoietin. Okay, then we still distal convoluted tubule. You got connecting duct, connecting duct, connecting duct into the collecting duct. Right? So keep doing that over and over. You guys are going to own this stuff. So you got Bowman's capsule, PCT, the loop, and the distal convoluted tube. Remember, the loop has a descending limb and an ascending limb. And at different parts of the nephron, you have different hormones that work, like ADH, antidiuretic hormone. That's going to work right here at the collecting duct. Antidiuretic, it's going to allow you to hold on to urine. So ADH is going to work right on the collecting duct. You're going to have things like calcitonin. Calcitonin is going to work at the distal convoluted tubule. Calcitonin. You're going to have Lasix, like your diuretics, your loop diuretics work over there at the loop of Henle. So the PCT, the loop, and the distal convoluted tubule, just review those main parts. We spoke about filtrate. We spoke about the collecting ducts. We're familiar with the papillary duct and the minor calyx, major calyx, renal, pelvis, ureter. We know that entire pathway. I had mentioned that the proximal convoluted tubule is made up of simple cuboidal epithelium, as is the distal convoluted tubule, simple cuboidal epithelium. But if you look at the loop of Henle, the descending limb and ascending limb, you got simple squamous. So where you have this at the loop, it's a lot easier for the diffusion of salt and water here. Salt and water to be exchanged at that loop of Henle. We know the difference between reabsorption and secretion. Reabsorption is returning the useful organic nutrients that enter the filtrate. It's returning it back to the body. Secretion is when you're trying to get rid of it all, right? Secretion leads to excretion. Much of the filtrate is reabsorbed, especially water, glucose, amino acids, and ions. It's reabsorbed, it's pulled back into the body. The secretion, that's going to help to manage pH, right? Because of getting rid of hydrogen ions. And it rids the body of all the toxic foreign substances. The reabsorption, we can pull back sodium, water, glucose, amino acids, and ions. Some reabsorption is passive, but most of it is active. And most of the reabsorption takes place at the proximal convoluted tubule. That's where most and the majority of the reabsorption takes place. What's not reabsorbed? We do not want to reabsorb urea, uric acid, or creatinine. We don't want to reabsorb that. Okay. We want to be able to get rid of and secrete hydrogen, right, to manage the pH, some potassium, creatinine, and toxic drugs. Okay, we went through the formation of urine, and this again, now that it makes a little bit more sense to look at that nephron, we can see what's in the filtrate. We can see reabsorption and secretion. They're indicated by the different colors. So we can see the poisons and hydrogen we're going to, and the drugs we're going to secrete so we can excrete. But our body wants to reabsorb, some glucose, amino acids, some salt bicarbonate, right? It's going to reabsorb some of those. There's your proximal convoluted tubule. Here's your distal convoluted tubule. There's your loop of Henle. And again, to review the basic kidney processes: filtration, reabsorption, and secretion. What is filtration? Whatever's in the blood and it pulls out into the proximal convoluted tubule, that's filtration. When it gets pulled back into blood supply, that's your body reabsorbing it. If it doesn't want it in blood, we're going to secrete it back into the tubule so we can be excreted. Now, just to show you, look at this. Look at how much water is being filtered. And look at how much your body is reabsorbing, meaning it's returning it to blood. Like a lot. How much are we getting rid of throughout the day? Maybe 1.5 to 2 liters become urine. Look at all the protein. We're filtering about 2 grams. We're reabsorbing almost all of it. We're getting rid of very, very small amount. But look at creatinine, 1.6 grams. How much are we reabsorbing? You don't want any of that. You want to get rid of it all. Okay. Look at glucose. Filter 162 grams. We reabsorb. We don't want to be peeing it out. If we're peeing it out, that's a pretty good indication of diabetes. Hormonal regulation of tubular reabsorption and secretion. Okay, these are the hormones that are going to be affecting different parts of the tubule. So angiotensin angiotensin 2, we know that angiotensin 2 is involved in retaining sodium and retaining water, so blood pressure goes up. We know that aldosterone is also involved in the retention of sodium and reabsorbing water, so that blood pressure goes up, right? All of these are going to increase pressure. ADH, anti-diuretic hormone, that's going to work at the loop of Henle. That's going to allow you to hold on to water again so blood pressure can go up. ANP, atrial natriuretic, that's going to do the opposite of these. It's going to get rid of sodium. It's going to decrease sodium, which decreases water. If you're not holding on to salt and water, then you decrease blood volume and blood pressure. Then you have PTH, parathyroid hormone. That is going to stimulate the opening of calcium channels at the DCT, distal convoluted tubule. Increase the reabsorption of calcium. So PTH and calcitonin are going to work at that DCT.